Breast cancers that do not express estrogen receptor, progesterone receptor, and EGF receptor or HER2 cannot be targeted by many of the common treatments for breast cancer, so are of particular interest. We can use TCGA clinical metadata to identify so-called triple-negative breast cancer or TNBC samples. To do this, we will create a sample set of all breast cancer tumor samples, but within this sample set, we will flag the samples that are TNBC. You will find under Clinical Data, Procedure, Genetic Testing, that many breast cancer samples were tested by in situ hybridization or immunohistochemistry for presence of ER, PR, or HER2. We will use these data to group our sample set. To create triple negative versus other breast cancer samples, first, filter for breast cancer, and filter only for primary tumor. Next, select only negative for estrogen receptor or ER, progesterone receptor or PR, and EGF receptor or HER2. The samples that are displayed are all breast cancer tumor samples that are negative for ER, PR, and HER2. Select these samples. Now, reinstate the filters for positive samples for ER, PR, and HER2. We now see all breast cancer tumor samples that were either negative or positive for ER, PR, and HER2, but the triple negative samples are still selected. Choose Create Sample Set, Group Sample Set from Selection. Name the sample set, and apply one or more tags. Under the Metadata tab, you can see that a sample set is a list of sample IDs along with additional optional metadata, in this case, whether or not they were selected. The selected samples were triple negative breast cancer. To use your new sample set, you may need to reconnect to TCGA land. You will find your sample set listed by the name you gave it,